Hi everyone, so in this video we'll talk about genomic library. So let's get started. So what is genomic library? So a genomic library is an organism specific collection of DNA covering the entire genome of an organism. So this particular library or this particular genomic library consists of all the genomes of a particular organism. So a particular organism can have different genomes depending on different species, but this particular library consists of all the types of genomes of a particular organism. So it is very much organism specific and contains everything what an organism consists of. So it contains all the DNA sequences such as expressed genes, non-expressed genes, exons and introns, promoter and terminator regions, as well as intervening DNA sequences. So it consists of everything what an organism consists of. So moving with that, so talking about the construction of genomic library. So a particular genomic DNA library involves isolation and purification of genomic DNA. So it all starts with a starting material, which is the DNA. After that, fragmentation of genomic DNA by physical methods such as mechanical shearing, sonification and enzymatic methods are followed. So that particular DNA fragment is cut into pieces by different restriction endonuclease enzymes on that particular DNA site so that we get different fragments, and thereafter these fragments are treated physically or mechanically by certain methods such as sonification, enzymatic methods to break them into fragments. So next up is the cloning of the fragmented DNA using suitable vectors such as lambda, EMBL4, cosmid or PAC. So after that we have got small little fragments of DNA or small little fragments of the gene of our interest. We try to clone them. So we'll try to clone them using a vector. So the some of the vectors that we generally use for cloning is BAC, cosmid and lambda EMBL4. After the cloning has been done, we screen the library to identify our clone of interest using a procedure that discriminates between the desired clone and the all others. So after cloning, we need to check which all are the ones or which all are the recombinant ones and which all are the ones which are not cloned or which are the ones which we don't require. So there's a very thin line between the two and we need to find out the ones that we need and the ones that we don't need. So these are techniques used for screening such as Southern blotting, hybridization, PCR and immunochemical techniques that are used for screening purposes, for screening of desired gene of interest after cloning. So thereafter a library is created containing representative copies of all DNA fragment present within the genomes. So after that a particular all of the selected ones after screening are put into that library, and this is how we say as genomic library. So this is a pictorial view. As you can see, this all starts with the DNA. As you can see, this is the DNA. It contains restriction sites of different endonuclease. So this is the restriction site, this is also a restriction site. So when these sites are attacked with the restriction endonuclease enzymes, these lead to formation of small fragments. As you can see, there are various number of fragments that are formed due to the presence of different restriction endonuclease enzymes. So as these fragments are formed, these fragments are ligated into the cosmid for cloning purposes. So as we know, we have got different fragments. So these fragments are used for cloning in different plasmid vectors or different lambda EMBL vectors or cosmid or PAC vectors. So in this case we are using a cosmid vector for cloning purposes. So after cloning we need to screen it. So how do we screen it? So in this case it's shown that we we try to infect a E. coli bacterium to screen that. So how it's done? So when we ligate those particular fragments into the cosmid and we get clones out of that, so we test that clones via infecting E. coli, and via infecting E. coli will get the colonies for the desired gene of interest. So you can see here, you can see some colonies here, and these colonies are then added to the gene library. So this is how the gene library or the genomic library is prepared. So this is very simple. As you can see, as we ligate all of the fragments into the cosmid, so these are cloned as per convenience, and these clones are checked whether the ones that we need and the ones that we don't need. So how do we check that? So it's a very simple method, as said. So we check it by infecting or using those particular clones in a E. coli bacterium. So we use those clones in E. coli to infect it. So we use it against E. coli, and the colonies that we get after incubating for, let's say, 24 hours or 48 hours, and the colonies that we get, we add those colonies into the gene library. So this is how we prepare a genomic library. So talking about the advantages of a genomic library. So it helps in identification of a clone encoding particular gene of interest. So as we know, these libraries consist of all the recombinant ones or all the clones that encode a particular gene of interest. So it's a library that consists of all the genes that are of interest. So we can identify any of the clones that we require in that particular library. So genomic libraries from eukaryotic organisms are very important to study the genome sequence of a particular gene, including its regulatory sequences and its patterns of exons and introns as well. And some of the disadvantages at the same time will be genomic libraries from eukaryotes have very large genomes containing a lot of DNA which does not code for proteins, and also containing non-coding DNA such as repetitive DNA, regulatory regions, which make them less than ideal. So it also contains, as you can see, for eukaryotes, it contains a lot of DNA that do not code for proteins. So these are not very much useful for proteins, and also consist of many non-coding or unnecessary regions such as repetitive DNA or duplicates that we don't need. Also, genomic library from a eukaryotic organism will not work if the screening method requires the expression of a gene. So this particular library also consists of cells that will not work if the screening method requires the expression of a gene. So the expression of a gene is not possible for the genes present in the or for the clones present in the genomic library. And talking about the last part, which is the application of genomic library. So talking about the application, it helps in the determination of the complete genome sequence of a given organism. So it helps to determine the complete genomic sequence of a given organism. Also, it helps in the study of genetic mutations, and it is used for genomic mapping, sequencing, and the assembly of clone contexts. So these were some of the applications related to genomic libraries. So let's just keep this video till here. Hope you enjoy this, and thanks for watching.